too, including this one. This one's fun. This one's fun. It's a good reminder that washing your hands sometimes just isn't enough. Just isn't enough. So why isn't washing food, or you could say why isn't washing our hands, or why isn't brushing my teeth, or why isn't washing the floor or the toilet or anything else? On Friday we had some very vivid imagery, so I'm going to try to keep that going today. With water enough to prevent infection. So this is something that came up with in our question where I said, why are extremophiles so interesting? And I mentioned that a number of bacteria make biofilms. So you need to remember that bacteria oftentimes make biofilms, and we'll talk a little bit more about what that is, how it works. But so much so that you can fossilize these biofilms. They are incredibly robust structures. Okay? And so what these bacteria are doing as they make these biofilms is they are secreting a sugar, a very sticky polysaccharide that basically form an extracellular matrix to hold that bacterial cell and 7 million more all together onto whatever surface they've made this biofilm on. Would it be your hands, your teeth, your toilet, your plate, right? Whatever they've made that biofilm on. So it holds them all together. It anchors them down. It anchors them down. On top of that, oftentimes, some of the pathology associated with the infection is due to that biofilm and not to the individual cells. Not to the individual cells. So this is kind of fun. It's not fun at all, actually. I lied. So here are some pathologies that are associated just with the biofilm and not actually the individual bacteria. So cystic fibrosis is basically it's a pump problem. There's a protein that acts as a pump to send out chloride ions, and then water follows. Okay, but if that pump is malfunctioned, the cells can't send out chloride. This is more than you need to know. I'm just giving you a picture. Cells can't send out chloride. Water can't follow, and mucus just gets really thick because you can't add any water to it. It basically turns into cement. And so places in your body where you rely on a nice, healthy mucus covering, you don't have that. And so that's like your respiratory tract, your bronchi, your alveoli, and your lungs, in some places of your digestive tract. And so bacteria build up. And then the bacteria start making a biofilm that just gets your immune system to react to their presence. And so on top of having a really thick cement lining those tissues instead of nice mucus, and you're like, why is mucus nice, right? But it is when it's in its proper place, right? On top of that, you have bacteria by the tens of millions that are also making biofilms that contribute to the pathology. Legionnaires disease is a type of pneumonia that happens as a result of bacteria in your lungs making a really complex biomatrix. Otitis Media, which is a type of inner ear infection it's not an inner ear, sorry, this is a middle ear infection with bacteria making a biofilm that just basically locks things up, induces inflammation. And then dental plaques are also what form on your teeth. Those are a biofilm from bacteria. On top of this, biofilms make bacteria extremely difficult to remove from food items, which is especially an issue for food items that aren't cooked before you eat them. So this is especially common for vegetables. Usually when you get some kind of like an E. coli or a salmonella outbreak, it's almost always lettuce or some kind of vegetable that is just typically rinsed with water before used in food. Okay. Any questions? Yeah.
The biofilm, it isn't necessarily exclusive to like bacteria, right?
I mean, bacteria are not the only organisms that make a biofilm. Other colonial organisms do as well, but bacterial biofilms are somewhat unique, if that answers your question. So they're not the only organisms that make biofilms, but bacterial biofilms are somewhat unique. Yeah. So some of these common biofilm creating bacteria that can lead to issues. I mentioned Salmonella, E. coli. Again, you tend to, I don't know, once a year, maybe twice a year, hear about some E. coli or Salmonella outbreak. And it's almost always either lettuce or onions, you know, vegetables that are washed but not cooked. Here's one, Clostridium botulinum, which is a bacterium that makes botulinum toxin. It's the causative agent of botulism. And so you tend to have this happen in canned goods that aren't properly sealed. And so the bacteria gets in, it thrives in kind of that low oxygen condition, makes a really wonderful biofilm that you don't really taste, but then it's just packed full of this toxin. Were you going to ask, is that where Botox comes from? Because it absolutely is, right?
To backslide just a little bit back to the E. coli.
Yes.
I believe it was this year that CDC did a recall on a bunch of cereal brands.
Interesting. Yeah, it's another food item that you don't cook, but the way it would come there is just contamination in some way. And you're like, how does cereal get contaminated with E. coli? You don't really want to explore that question. Because E. coli contamination usually comes from fecal-oral contamination spread. And so if enough cereal to have to recall a brand of cereal was infected with E. coli, it's kind of an issue, right? Vegetables, it kind of makes sense. You're like, you're watering this with water, right? And you're like, oh, we use gray water because it's wonderfully efficient, right? It's responsible. But sometimes gray water is not gray water. Sometimes gray water is black water, right? And if you don't know the difference between gray water and black water, that will be a reading pleasure for you later today. Yeah.
What about an issue with cooked foods? Because there was also 2 years ago an incident where, like, some cooked chicken.
Oh, sure. Yeah, I mean, improperly cooked, or sometimes you just, it depends on whether it's the toxin or live bacteria. If it's toxin concentrating, sometimes it doesn't matter whether you cook it, the toxin is still. So the botulinum toxin, what it does is it prevents your muscles from relaxing, which is really awesome if you want to get like an injection on your eyes or your lips because you've got wrinkles and you want those muscles to contract and stretch out and not relax. Works wonderful. But when it's all over your body and your muscles can't relax, you've got a major problem. Yeah, major problem. Yeah, Morgan.
That's not the same thing like the Clostridium
Botulinum?
as like C. diff?
No. Same genus,
Okay.
Clostridium, but that's difficile.
That's what it's
Yeah, we'll talk about C. diff in a little. Same genus. Yeah, just wonderful species in that genus. That's a good genus. It's a very giving genus. Yeah, Joe.
How does it spread to the whole body, but they can be like exposed to like Botox or something?
Yeah, well, when you inject something subcutaneously, you can keep it out of the vasculature. So like if you put it directly into a subcutaneous muscle, it's probably not going to be absorbed in the bloodstream and not at enough to actually create some issues. Unless you're getting really, really extensive injections constantly and huge doses. The biggest problem is usually for people that are using Botox injections to treat types of arthritis. Those are where you have the most concern because you're using really heavy doses. Yeah, really heavy doses. All right, so here is a figure right out of your textbook. I know I've showed you some of these and said these are terrible figures. This is a great one. So absolutely, this is a wonderful figure. And it basically shows you the 5-part process of making a biofilm. So step 1, bacteria just kind of coat a surface. And at this point, you can wash them away fairly easily, but they're just kind of coating and they're aggregating. They're doing what bacteria do best, and it's really doing what living things do best. They make copies of themselves, right? And so they coat this surface. Would it be your teeth, your hands, your toilet, your food, right? They don't care, and there's lots of food there. They eat it. They make copies of themselves. And then step 2, they anchor themselves to the surface. They anchor themselves to the surface using the pilus. And we talked about that on Friday as a way of exchanging information, but it's just a tube. It's also a great way to anchor themselves. And then step 3, they start making a lot of that sugar. That's going to even adhere more bacteria than are just in contact with the surface. Because the ones that are in contact with the surface, they're anchored down. But then the bacteria that are on top of them, and the bacteria that are on top of that pile, and on top of that pile packing, you know, tens of millions in, they're all anchored because they start secreting a lot of sugar. And then in step 4, you start to have your biofilm growing and spreading out, right? It almost becomes its own entity. You know, millions or hundreds of millions of bacteria behaving almost as a single organism. And here you'll see it start to spread. Like if you, let's just say you wanted to do something and you're like, I'm gonna take one tooth and I'm never gonna brush it, right? Just never ever gonna brush that tooth. And then it'll start to build up that biofilm, and then you'll see it start to spread, and then you're brushing next to it, right? Because you're fighting that, but it's like this entity is spreading. And then in step 5, the biofilm starts reproducing. The biofilm is making new biofilms by different bacteria popping out and going and finding and settling and finding a new place. So it's not just the biofilm growing and spreading, but it's actually reproducing. It's sending out bacteria to go make new biofilms, maybe in somebody else's mouth, right? As a result of that one tooth that you didn't want to brush, right? And so you built this monster of a biofilm. Man, I really feel tempted to do that now, right? To just take one tooth and just be like, you know what? If I have to get a root canal on that one tooth, I'm going to do it for science, right? I want to see. Yeah, Joe.
How long does it take to go through all these stages?
It depends on the species. It depends on the species and how much food is available. If inside of your mouth, it won't take long at all, because there are several different species of bacteria that live in your mouth, and there's plenty of food all the time, right? Because they don't just eat what you eat. I mean, they eat what's in your mouth too. Like you make saliva, oh, sure, I'll eat that. Skin cells flaking off of your cheeks, yeah, I'll eat that too. Like there's plenty of food available, right? It's just a wonderful thing to think about. Wonderful thing to think about. All right. So here's just showing you vegetable sprouts grown at an organic farm, the cause of an E. coli outbreak that killed 32 people in Germany. But it usually happens a couple of times a year. In B, it's showing you a zoomed up version of this particular strain of E. coli. You see that nice rod-shaped, very characteristic of E. coli. And then C, showing you what happens as a result of the toxin of this particular strain. And basically, it destroys red blood cells. So here are nice, healthy-looking red blood cells. Look like donuts or discs. I prefer donuts because they're delicious. And then here, these are not nice, healthy-looking red blood cells. Many of these have been ruptured as a result of these bacteria. So, all right. Now we're going to transition from the evil of bacteria to the good, right? The other side of the coin. The other side of the coin. What positive role do bacteria play? Well, we've already mentioned this one when we said that bacteria are an integral part of the cycles of a number of different nutrients. If bacteria could not pull nitrogen out of the air and make it available to plants, life would not work. Bacteria are the only organisms on Earth, as far as I know, that can pull nitrogen out of the air and make it and convert it into a form that plants can actually use and fungi can use. And then we eat plants and fungi and then we can use it. But we can't actually take atmospheric nitrogen and change it into a form that we can use. So without this, life wouldn't work. So what positive role do bacteria play? They make life possible, right? That's kind of good, right? You're like, okay, I'll allow them to live in my mouth so that I can survive. I mean, you don't need the anyway, actually you do need the bacteria in your mouth. I actually said you don't, but you do. You need them there. Almost as important as life wouldn't be possible without bacteria, cheese wouldn't be possible without bacteria. Right? It's like life, and then cheese is right there. Just right there. Maybe closer. And so we have this whole industry called biotechnology, and a big chunk of that industry is figuring out wonderful, novel ways of using bacteria to make new materials, foods, textiles, all sorts of wonderful things, right? Yogurt, cheese, bread, okay, just all these wonderful, wonderful things. The textbook also talks as beer and wine, but those are not wonderful things. Those are evil things. All right? Bioremediation. Bioremediation, this is another wonderful thing that bacteria can do. Right? We have a tendency to just mess everything up. Maybe it's just me. Well, it's not just me. But maybe you're not in this boat as well. But I have a tendency to just mess everything up that I touch, right? I'm like the opposite of King Midas. You know, King Midas, he touched something, it turned to gold. I'm like the opposite, and oftentimes we do that. And so there's one thing humans are really good at. There are actually two things humans are really good at. Making new humans and making a mess of everything else, right? And so the nice thing is bacteria can clean those messes for us. This is called bioremediation. And so, you know, like there's mercury contaminating the soil. I wonder if there's a bacterium that eats mercury. And what do you know? There is, right? And so you just sprinkle those bacteria on, they eat the mercury, they clean it up, right? They convert it into something that's less toxic, right? You have a massive oil spill. You know, like, I wonder if there's a bacterium that can eat oil. And you find not only is there one, there are dozens or hundreds of different species. And you just sprinkle them and they eat the oil into something that makes it water soluble. Because that's why oil is so hard to clean, because it's not water soluble. It doesn't dissolve in water. Right? So you got to use Dawn. You got to take that little baby duckling covered in oil, and you got to get Dawn, because you just can't use. It's not funny. It's horrible. What's wrong with you? You can't just use water to clean. To clean it, you have to, right? Because it's not water soluble. But bacteria will eat that oil and convert it into something that is water soluble. It makes it a lot easier to clean up. And then there was this would be this was like maybe 15 years ago. There was there's a nuclear power plant in southeast Washington, and they were burying the nuclear waste, which was wonderful until it started leaking, which was still like, oh, it's southeast Washington, it's not heavily populated. If you're from southeast Washington, you know that, right? It's just not a really densely populated area. But then it got into the groundwater, and it was moving to the Columbia River that separates Washington and Oregon. And you're like, oh, well, that's not that big of a deal. It's the highest volume river in the world. It moves more water than any river in the world. And it was just it was headed towards that river. And then so there were people like, I bet we can find bacteria that will eat this nuclear waste. And they found some. And they built little nets that the groundwater had to filter through, full of bacteria that would eat the nuclear waste. And because it was just a small amount of nuclear waste moving through the groundwater, they were actually able to remove it before it got to the Columbia River, right? Big win. You're like, well, why don't we just do that with all of our nuclear waste? Because there's a lot of it, right? And it takes a lot of bacteria, yeah.
It's happening at the WIP isolation plant in New Mexico.
In New Mexico?
Yeah.
Do they use bacteria to clean it up?
They've shut it all down.
Yeah, they shut it down. What? Yeah. I mean, it's like just like we make a mess and the bacteria will clean it for us, right? And it's just wonderful. On top of that, we have billions, literally billions of prokaryotes, if not more, making up what we call our microbiome, what lives inside of us. Without these normal flora, things like, here's Clostridium difficile, C. diff, grow out of control. And if any of you have ever had C. diff or know somebody that had C. diff, it's not great. It's not great. Right? You got to have like your own toilet, like your own area, because it spreads like wildfire. Yeah, Mia.
So back on like the bacteria,
Yep.
using the cleanup stuff. Is it like possible that there can be like negative consequences of using that? That like like when they find a bacteria, like it actually can have like a negative impact.
Sure.
Like it could like leak into the river, and like it can make people sick.
Yeah, it's kind of like when it's kind of like when you accidentally introduced an animal to a new space, right? Like we accidentally introduced rats to an island. Like no big deal, except for now the rats are eating all the chicks of all the birds that live on the island. You're like, I know what we'll do, because we can't get every single rat. We'll introduce cats to the island to eat the rats, right? And this is kind of like what you're doing where you're introducing bacteria to clean up your spill. Like we'll introduce cats to eat the rats, but then you realize, wow, the cats, they love to eat these island birds because the island birds have grown with no predators, and they just, like, they don't run away from cats. Like they just, I mean, it's just wonderful. So instead of the cats eating the rats, now they're eating the birds too. And it's just like, now you're like, okay, well, what are we going to introduce that's going to eat the cats and the rats? Or you can just do what Florida does with their boa constrictors, and you're like, we'll just pay people to come and to kill them. Do you know that Florida pays you to go and kill boa constrictors? In case you were like, man, I really need a summer job. You can go and take on 15, 16, 20-foot boa constrictors, and it's actually decent. It's decent money. It's hard work. It's hard work, but it's decent money. In case you were, thinking about what you were going to do this summer. All right. Any other questions, thoughts, concerns, things you want to share about bacteria? Because we're going to move from chapter 22 into chapter 23, and we're going to leave bacteria for a little bit. I mean, you won't leave bacteria because they're all over you and inside you, but we're going to stop talking about them for a little bit. You will never, ever stop thinking about them. All right? Okay, so we're going to take a lecture break, and as we do, I'll transition over to chapter 23. And so here's kind of, hopefully, the picture you're starting to get for many reasons. The picture you should have is that if there is any environment on Earth, bacteria are going to live there. If there's any possible thing to eat, there are going to be some bacteria that eat that, right? That's hopefully the picture you're starting to get. Okay, and so what I want you to do, talking with those around you, I want you to readdress that question we talked about on Friday, is how do we get that sheer variety of bacteria to where you find them anywhere on Earth, any environment that you can imagine, eating any type of food you can imagine. How do we get just that sheer variety of forms? But this time, you don't have to get them all from a single extremophilic bacterium. You can use anything at your disposal. Anything at your disposal to explain just the sheer variety of bacterial forms that we have. Okay? Any tool you can imagine. Does it make sense? Do we know what we're doing? And remember, keep in mind, I like these questions, right? And I don't just like them because I asked them, but I like them because they make wonderful exam questions. All right? Take a couple of minutes, starting now.
I would say maybe eventually maybe the bacteria that's producing or maybe yeah, they're able to change
Saying that there's like a variety of sources, like host sources, but there's also a variety of foods they can take, so they can take on so many because they're easily adaptable.
I think it also has to do with the variety of environments. If you think about like the environment in the Amazon rainforest is very different from the environment so the bacteria in the Amazon so yeah, I also spend I think the environment has to see that.
yeah.
Alrighty. So what are some ideas? Amanda.
Because of the different types of environments that we have on Earth, like especially at the different latitudes, like places at the equator, you would find different bacteria than you would find here.
Absolutely. Yep, absolutely. So the reason why we have such an enormous variety of bacteria is because we have such an enormous variety of ecosystems. right? You ever heard the saying like it's a dirty job, but somebody has to do it, right? I'm pretty sure that, bless you, that saying actually came from a microbiologist studying bacteria, right? I mean, that basically just sums up the way bacteria live their lives, right? Humans, we need help, right? We need help digesting lactose, which is a sugar that you find in cow's milk and other milks, right? Because we need to eat cheese, right? I need to eat cheese. If you don't eat cheese, that's fine. But I need to eat cheese, right? And I need some way to be able to digest that lactose. And so it's a dirty job, right? Something's got to live inside my intestinal tract and help me digest that lactose. It's a dirty job. Somebody's got to do it, right? And the bacteria do it. Any other thoughts? Yeah, Joe. And then we'll come up to Mika.
Evolution, not Darwinian evolution.
Okay.
Evolution, like over time and adaptation.
Okay. So that you started with a, so you're getting at, you started at a point where you didn't have as much variety, and then through just kind of diversification, you developed new forms. Okay. Interesting. We're going to come to that. We're going to spend like several weeks of class talking about this very idea of how much diversity is reasonable to assume was created and how much diversity has come after the initial creation. Yeah, Mika.
I was thinking like they would mutate so that they would like fight antibacterial.
Okay.
In terms of like bad bacteria.
So you got a mechanism of, I mean, just mutating to change some characteristic about them. Interesting. Okay. Any other thoughts? Yeah, Mia.
So is there like bacteria that can live in you other than like enzymes that can like break down like?
Oh yeah. Oh yeah. I mean, there are bacteria that live in you that you have no need for, but they live there just because they can, right? And they're not hurting you in any way. It's a nice, safe, warm place for them, full of food, and they're not hurting you in any way, but they live there because they can.
Are there like supplements like that like use bacteria instead of like the enzyme?
Oh, absolutely. Yeah. So most probiotics use live bacterial strains. Yeah. So a lot of probiotics use live bacterial strains. You know, like you've got probiotic yogurt. I mean, yogurts, oftentimes you've got the biofilm. I mean, look at that, kombucha.
I'm drinking it right now.
Yeah, she's drinking it right now. Nice probiotic with live bacterial strains. You know, and some of it is bacteria that her body needs. Some of it is bacteria her body doesn't need it, but that bacteria need a place to live, right? They need a place to live. Yeah. Allison.
Oh, I was just going to say that because the life cycle of the bacterium is so short,
Yeah.
there's opportunity for a lot of mutations, whereas like in the human population, you don't see that.
Yeah. Yes. Yeah, it doesn't take long to get through a bacterial generation. For E. coli, it's every 20 minutes. Every 20 minutes is a full generation, which for humans, it's on average 30 years, right? So you're accomplishing 30 years worth of time in 20 minutes. It's what allows us to, in the last, couple of decades, we have studied the equivalent of hundreds of millions of years of human evolution potential inside of bacteria. I mean, their genomes aren't as big as ours, but, what would equate to hundreds of millions of years of time. Yeah. And then there's, it's undoubtedly there's some of it that was created, some of this diversity that was put in place because it was necessary, right? It really changes the whole idea of what does it mean to be human, right? Can you actually be fully human without the gut bacteria that help you digest what you eat? And that's a very interesting question, because if the answer is no, then it has to be you were created with those bacteria, meaning you were created with the sense that some of the cells in your body are not genetically identical to each other. In fact, are so different that they'd be classified as an entirely different type of organism altogether. But your body needs them to survive. And then so it's like, well then, with heavy antibiotic use, are we making people no longer human because we kill all their gut bacteria? I don't know. I don't know how to answer that question. Autumn.
How did the gut bacteria get there?
That's a wonderful question. That is a wonderful question. Some of them get there in utero. So they're a gift that your mother gives you along with life. right? They come as like a package deal. Many of them come as a result of eating in similar ways to other people. So what you find is that the gut bacteria you have are more similar to the gut bacteria of your family than they are to anybody else's gut bacteria. And so part of that is because you got them in utero, but part of it is because you have a very similar environment to your family's environment. And then what you find is during like the school year where you're away at college, your gut bacteria start to merge with your roommate's gut bacteria. They start to be more similar because now you're in a different kind of environment, right? All eating at the same calf, right? Brushing your teeth with toothbrushes that were exposed to the same environment, right? It's so wonderful. So you actually, every single day you spend with your roommate, you become more like your roommate on the inside. On the inside. This is fun. So these are fun conversations to have. These are fun conversations to have. All right, chapter 23. We'll stop talking about bacteria for a while. You're like, yeah, you're right. We're becoming more like Christ together, right? And that's true. But you're also becoming more like each other in your bacterial microbiome. All right, so how does structure and reproductive strategy vary with environmental stability? So we're going to start talking about eukaryotes. Now we've got cells. They have nuclei. They have membrane-bound organelles. These are arguably more complex cells. Okay? And so how do these cells actually vary what they feed on, how they look, and how they make copies of themselves based on their environment? Do eukaryotes evidence universal common descent well? And then we'll start getting into more specific questions, but we're not going to get to there today, so I'm not going to read them to you. I'll read them to you later. All right, so this first one. How does structure and reproductive strategy vary with environmental stability? In order to answer this question, first we have to figure out what on earth is a protist, right? This chapter 23 is all about protists. And any of you have a junk drawer? Even if you don't have a junk drawer, how many of you know what a junk drawer is? There we go. That's a protist, okay? Protists, they are the junk drawer. So anything that's a eukaryotic cell, if it's not a plant, an animal, or a fungus, it's a protist, okay? It's the junk drawer. It's like, we don't want to have a whole bunch of different types of eukaryotes. So what we'll do is we'll have 4, plants, animals, fungi, and everything else. It's like the miscellaneous drawer. That's the protist. So if that's true, and you've seen a junk drawer, you can imagine what a junk drawer is like. Would you imagine a lot of variation or very little among protists? A lot, right? Because like, what's in the junk drawer? Like pens, things your kids bring home from school that they want you to hang up, and you're like, oh, I'm going to hang it in my office. And you put it in the junk drawer. I'm just kidding. That's so cruel. I don't do that. I hang it up. I hang it up in Dr. McLean's office. So that's also not true. I'm lying to you. I apologize. I thought it was funny. It's pretty funny. Yeah, and so what you find, there's an enormous amount of variation. Enormous amount of variation among protists. I mean, it truly is the junk drawer of eukaryotes. So you got some that live in nice, stable environments. You have some that live in extreme environments, and those that live in extreme environments have shells or some other non-living material on the outside, a pellicle, to protect themselves from just drought, from intense heat, from intense pressure. And all of you have had this lab now, and so you've seen some of the variety. Dinoflagellates are protists, diatoms are protists, amoeba, and I know we didn't have the living amoeba, but we had the prepared slides. And just an enormous variety of forms. Enormous variety of forms. Also, an enormous variety of metabolic strategies. Although most of these include something that you very rarely find in bacteria called phagocytosis. And this is the process of eating something really large. Either an entire cell, or at least something really big that you can't just transport across the membrane. You actually have to surround with your cell and pull inside. It's a wonderful way to feed. It's a wonderful way to feed. Allows organisms to eat objects that are sometimes larger than they are. Just stretch their membrane around it, and then start breaking it down. So on top of this, you have some species that are called mixotrophs, and they entirely change what they eat on based on the environment, what they feed on. Eat on, I guess that works too, but it sounds weird. What they feed on based on the environment. And we are mixotrophs. Like, if you're in the calf, you eat what's in the calf, right? If you're in somebody's room and they've got a bowl of chips, you eat the bowl of chips, right? Or if you find something that looks delicious in some random refrigerator you don't know who it belongs to, you eat that, right? Just like you eat what's available to you. Is it just me? Gosh, I'm having fun today. I hope you're having fun. All right. Now, most protists are capable of a pretty extensive movement. In response to some stimulus. This is one of the characteristics of life, is that the organism must respond to a stimulus. Now, protists, many of them are mobile, and so the way they respond to a stimulus is to move toward it or to move away from it. Move toward it or away from it. Why would you want to move towards something? What's the stimulus that you would want to move towards, Levi? A food source, right? Some chemical stimulus, right? Like a peanut butter cup. Like, I smell that, and I want it, right? Yeah. What's that? Your dog? And so what stimulus are you picking up on? Like a visual stimulus, or is it yiping? Is it a yiping dog? Okay. She what does what? She cuddles. Okay, so a visual and a tactile stimulus, right? So actually, mammals are somewhat unique in our ability to actually sense comfort from a touch, right? To actually immediately, and it's not just humans, it's most mammals, and that's somewhat unique among animals, right? And so you've got a touch stimulus. Okay, I heard somebody up here say light, but then they didn't raise their hand, they didn't want to re-say it, but light, right? Sometimes light is a great stimulus to move towards, because in the light are things to eat, right? Things that are taking sunlight and converting them into sugar, and then you're like, there's just this bag full of sugar, and I want to eat it. Right? And you can. What are some stimuli that you would want to move away from? Heat?
Darkness.
Darkness, right? The absence of light.
Any kind of threat or danger?
Yeah, so a number of stimuli that you could use to perceive that. Auditory, visual, chemical, right? Like a skunk sprays, and that is a stimulus that you would like to move away from. I want to tell you something, and it may completely change the way you feel about coffee. The next time you smell coffee, I want you to think about a skunk spray. And notice how similar the two scents are. Yeah.
Predators.
Yeah, predators. Okay, and so what's interesting is the stimuli, your response to it is going to vary, right? If you're an organism that needs sunlight to create, like that's where your energy source comes from, you have to move towards sunlight. But if you're an organism that you don't need sunlight to get your energy, and being in the sunlight exposes you to predation, you may move away from the sunlight, right? Think like a cockroach, right? They move away from the light, which is, it's kind of fun if you are, you know, in the game of training cockroaches. It's a wonderful stimulus to provide negative reinforcement. That's another story for another time. So sexual reproduction is incredibly common among protists. It's rare among bacteria, but it's very common among protists, and it usually happens in very variable environments, like a temperate freshwater environment. And I mean temperate as not tropical, and so temperate as in like a place where fresh water freezes over in the wintertime, right? And you're a protist and you move around in water, and frozen water is very difficult to move around in. Would you agree? Yes. Okay, so we're on the same page. And then so one way to actually deal with that is you may actually, you know, prepare the next generation and form a cyst around like this cell just to keep it alive, and then you die, right? You prepare your offspring to actually emerge when the ice thaws, and then you die when the lake freezes over. And so you get to make copies of yourself and set those copies up for success, right? Let's get them through the winter, and then they'll emerge, and it'll be spring, and there'll be lots of wonderful, things for your offspring to eat. All right? So sexual reproduction, again, very common in produce. On top of it allowing you to actually take offspring and coat them with something that will allow them to survive, it also allows you to mix genetic material, right? So you may be thinking, man, my body struggled at the very end of the growing season. Maybe if I was able to combine some of my genetic information with another individual's, my offspring would be better equipped to handle it than I was. And the beauty of it is you don't have to think that way in order to behave that way, right? Organisms don't have to consciously think, man, it'd be nice if I could mix everything good about me with everything good about another individual and produce something even better in order to behave that way. Because individuals that do that and make more viable offspring are going to have more offspring that survive to make their own offspring. So wonderful. Like, they don't have to consciously think that way in order to behave that way. So here's an example of phagocytosis. Here's just, I don't know, some cartoon protist, and it's wrapping pieces of its cell around something really large, too large to just move through the membrane. And so it wraps around it, creates a little vacuole, and then that fuses with another vacuole containing digestive enzymes, breaks down it, releases a lot of stuff as food. Anything non-eaten will, not eaten. What is that? Not eaten will be extruded as waste. You're like, cool, it's a little cell carrying out all the activities of life, including eating and defecating. And you're like, wow, it's really awesome. You literally are carrying out all of the most important aspects of life in a single cell. Right? It's beautiful. It's beautiful. All right, so when we were talking about bacteria, the only means of locomotion that we talked about were flagella. Do you remember that? We had a slide, and I'm like, look, there's a bacterial flagellum, and that's, if you have a mobile bacterium, that's how it's going to move. Flagella are nice for small cells. Flagella are not nice for large cells, okay? And so large cells have a really difficult time moving with flagella, and so some protists are really large, and so they can't move with flagella, so they move with cilia, or they move by just kind of shoving their whole body in a particular direction. This is what I really wanted you to see, but the company that gives us our living materials, they didn't send us amoeba. But it's wonderful. It just basically like throws fluid at its wall and just rolls in that direction. It's wonderful. It's wonderful. It's beautiful is what it is. But you didn't get to see it, but here's a cartoon of it. And then here's an example, just kind of a characteristic type of sexual reproduction found in protists. This specifically is paramecium, but you find similar processes in other protists. Yeah, Chris?
Can eukaryotes still use a flagellum if they're small enough?
Yes. Yep. You do find flagellated protists. So dinoflagellates, which you looked at in lab, they're called dinoflagellates because they have flagella, but they are pretty small. And then the trypanosoma that you looked at the very end of lab that are so small that it's like really hard to see a single individual, those are flagellates. They have flagella. But yes, but small protists can move with flagella. Larger protists that you can actually see fairly well, even under like medium or scanning power, those are not going to use with flagella. They're going to move with cilia or with something else. So here, you actually, in order to have true sexual reproduction, you have to combine genetic material. And so that's what's happening during this. This is called conjugation in paramecium, and they are actually exchanging genetic material. And so the next generation, which actually happens along the same generation, it's just one completely conjugates the other. Like this one gets rid of all of its genetic information, or most of it, and then takes on some from another individual. And so you actually get a nice exchange of, bless you, of genetic information. Yeah. All right. Okay. Any questions? How y'all doing? this is a great day. It's a great day to be alive. I woke up this morning, and actually I didn't because I didn't go to sleep, but I would have woken up this morning, and I just knew. I knew that there was going to be something good because we left off on such a good note on Friday, and then we just get to keep like these wonderful thoughts just moving forward. That one of like you and your roommate, I mean, that just came to me. That one like just came to me as I was standing here. I didn't even do some thinking ahead of time. You're welcome for that one. All right. And also, you're like, that's fine. I live off campus. It's totally cool because you're still mixing a lot of your environment with other people. Yeah, it's just wonderful. Coworkers, classmates. Yeah.
So, as an extension of that, if you have pets, are there?
Oh yeah. Oh yeah. I mean, not like the full suite of bacteria can't make that transition, but there are many that do. Oh, absolutely. So yeah, you become more like your pets all the time as well. On the inside, right? You're like, it's awesome. All right, so we've got one more question to deal with today. We've got lots more to deal with this semester, but one more today. Do eukaryotes evidence universal common descent well? No. And yeah, some of you have already seen the slides, and the answer is absolutely not. I'm going to show you something. So I mean, that's just a very simple answer. It's no. You know, so if all you did was looked at how they look, it's already a mess, right? It's like I told you, it's a junk drawer, right? You just like open up a drawer, you're like, this isn't an animal or a fungus or a plant, it's going to go in there. And then the next thing you know is there's just a huge variety of different materials in there, right? And there's no particular order to it. But then when you add genetic information, it makes the story way, way more complicated even. Way more complicated even than when you just use morphological data, their structure. And so what you find is, and this is a term we're going to see a lot in this chapter and following, this idea of convergent evolution. And the idea here is that there are certain requirements that an environment places on an organism. And so if you're going to survive in that environment, you need to figure out a way to make use of those resources. And because of that, it starts to, again, lead to some similarities, right? Like with pets. Like if you feed your dog from your table, even just a little bit, you and your dog are making use of the same resources, and your dog starts to be more like you on the inside, right? Its gut bacteria is going to start to be more like your gut bacteria, right? And so in a way, bless you, the way you would explain similarities in the gut bacteria between you and your dog would be by convergent evolution. You had similar environmental requirements, and so both of you, although you wouldn't do that for, anyways, you wouldn't do that for single individuals because individuals don't involve, populations do. I know that. We'll get there. But this is kind of the idea of convergent. All right, and then I'm going to show you a new term, and we'll be done here, is this term polytomy. What does the root poly mean? Many. And then tomy is basically a representation of ancestry. And so polytomy is when you have a single ancestor split into multiple groups. When you have a single ancestor split into multiple groups. These are bad. These are bad if you want to have a good understanding of the history of that group. Because this isn't the way speciation works. Anyways, we'll get there. We'll talk more about this on Wednesday. But to sum this up, the reason why this answer is no is because when you try to group all eukaryotes together, you get just a big hot mess. A big hot mess. You could just write that. I mean, it's worse than a junk drawer. It's like taking a junk drawer and trying to route everything in that junk drawer back to a single item. Like, and how did everything in here come to be from just like a single item? Like, how did the paper clip I stuck in there become a pencil and a pen and some randomly partially chewed gum, right? And like a half-eaten sucker. These are things that are in the junk drawer in my house. Yeah.
What was polytomy?
Polytomy is when you have one branch or one ancestor split into many. Yeah, many branches. All right. Have a wonderful day. Enjoy the rest of your Monday. Be safe. Yeah.
So Ollie told me that